again, thank you all so much for inviting me to come speak to you. I have no conflicts of interest or disclosures, other than I guess I have a lot of passion for patients with heterotaxy. And so we're going to talk about and be really focused on how laterality is established. This is to give a foundation to better understand why patients with heterotaxy can have the spectrum of findings they do. Then we'll also review one gene and how it's been understood over time to affect laterality to sort of illustrate the complexity of the process. And when developing, there are three axes or sides that are defined. The process is generally thought to be highly conserved across all vertebrates and even some invertebrates. This fairly universal conservation means we've been able to learn a lot about these three axes from animal models. The anterior-posterior axis differentiates head from tail, and the dorsal ventral axis differentiates belly from back. The naming for these first two makes much more sense if you picture it from our four-legged relatives' perspective. And then the last one is the left-right axis, which this is the main axis we're interested in here. Though if either of the prior two axes are also disrupted, the left-right axis is disrupted too. The exact order of events is unclear, as the majority of this happens in the first 2 weeks of development. There have even been some researchers that suggest that the axes are established as early as the 2-cell stage. But evidence is still lacking as to what exactly is occurring and what the exact order is. But it's clear that both the anterior posterior and dorsal ventral axes need to be defined prior to left-right, as they're required to orient left-right. And so I'm going to try to briefly summarize the developmental process in left-right axis determination with a fairly generalized big picture approach. I'm keeping to generalities partially due to time, but also, as previously mentioned, there's still some uncertainty related to the exact details, but the majority of publications seem to be in agreement. And so the node, as we saw in our first talk, is a ciliated embryonic cavity established at the anterior aspect of the primitive streak, critical to multiple aspects of development. The node plays a functional role in establishment of laterality, though laterality is at least partially established by cellular signaling that happens upstream of the node prior to complete node formation or presence of cilia. And then both cilia within and surrounding the node further define laterality. Cells within the node are monociliated with rotary modal cilia that share configuration typically attributed to sensory cilia. The nodal cilia will begin to have a clockwise rotation, which begins to circulate fluids in the node from right to left. While the modal functioning of these cilia within the node break the symmetry in this action, it's actually the sensory cilia that are on the cells surrounding the node that result in the embryologic developmental changes. These changes in flows cause different gene expression at the lateral plate mesoderm and ultimately affect organ formation. And molecular genetic information we have supports the importance of the node and ciliary function to establish laterality. This is a critical part of research and understanding the process. When you can identify underlying genes guiding the process, you can then begin to alter models to both better understand that gene's function as well as get a better overall picture of the developmental process by seeing where specific disruptions affect specific steps and what the effects are. So we're going to dive into this a little bit more by focusing on one gene and the evolution of understanding and its role in heterotaxy over time. And so to illustrate some of the concepts related to how laterality is established, we're going to focus on ZIC3. ZIG3 is an interesting gene for a number of reasons. It's X-linked. It's still the only X-linked cause of heterotaxy we've identified. I'm not going to be able to do the story justice here, but as we're going to see, ZIG3 also has direct implications for node formation as well as ciliary function. This allows ZIG3 to be a unique research target and to learn about heterotaxy and establishment of laterality. Focusing on ZIG3 also allows me to focus a little on one aspect of my partner, Dr. Stephanie Ware's work in heterotaxy, which is an opportunity I'm always happy to take. Now, Dr. Ware's first paper on ZIG3 came out in 2002, but we're going to start with one a few years later in 2004. As an aside, in 2002, I was tweezer feeding pieces of baby brine shrimp to inbred jellyfish under a microscope, and I'm pretty sure all I understood about genetics was Punnett squares. So, but this paper describes 5 patients with ZIG3 variants identified within a cohort of patients with heterotaxy or isolated cardiovascular malformations, and also reviewed a few other published variants. Based on the location of these variants and what was understood about ZIG3's function at the time as a transcription coactivator, they were able to show their variants resulted in decreased transactivation of an SV40 luciferase reporter. This bar graph shows the different ZIG3 variants they tested across the bottom, and then shows the level of luciferase activity relative to the controls. They also looked for nuclear co-localization of this transcription factor and found that a number of their variants resulted in abnormal presence of ZIG3 in the cytoplasm of the cell. And so this first vertical pane shows the location of ZIG3 for each variant tested in red. The second one shows the nucleus for the same view in blue. And then this last pane is the overlapping. And so you see in the normal ZIG3 function, panel C up in the corner here, there's a complete overlap. And then the lower 4 pictures, F, I, L, and O, you see that there's representation of ZIG3 in the cytoplasm of the cells. And so we're going to skip a few years to a couple of 2006 publications. At this time, they've been working on a ZIG3 mouse model. They've shown that mice null for ZIG3 have a number of functional embryologic anomalies, including abnormal formation of the node. And so panel G here, right in the center, shows decreased expression of T at the node compared to the normal mouse embryo and F right above it. And so they also crossed the ZIG3 mouse with a nodal mouse model and found that based on those results that ZIG3 resulted in abnormal nodal expression fitting into that period of upstream of the node development of the left-right axis. Now we're going to time travel quite a bit into this 2013 publication where they're evaluating the timing of expression differences in ZIG3 and the ZIG3 mouse. They looked at mice with conditional loss of ZIG3 function. Since you can get isolated cardiovascular malformations with ZIG3 anomalies, they wanted to know if the effects on cardiovascular malformation were early or if they were part of the actual cardiac formation. So they made different conditional ZIG3 deficient mice that showed deficiency in cardiac tissue at different stages of heart development. And the results of this study suggested that all of the effects that ZIG3 had that disrupted laterality and heart development occur within the node and prior to cardiac tissue formation. And now we get to a 2018 book chapter where they're summarizing what's known about ZIG3 and the establishment of laterality. At this point, there's embryologic data from mice, frogs, zebrafish, and data from humans found to have ZIG3 variants. And both loss of function and gain of function of ZIG3 seems to disrupt laterality in animal models. Now we're going to look at this 2021 paper that looks at ZIG3's role in establishing laterality. This paper describes how ZIG3 helps position the rotary monocilia appropriately on the nodal cells. In the title, you'll see a term we haven't explicitly discussed, plantar cell polarity, which means the cells are organized uniformly across a tissue to asymmetrically express features in a two-dimensional plane, specifically the anterior-posterior axis. And they found that ZIG3 disrupts plantar cell polarity with multiple apparent mechanisms. For example, they found that when located in the nucleus, ZIG3 seems to work with RAC1, a gene known to regulate cytoskeletal elements of plantar cell polarity, and synergistically transactivate elements to establish the axis. They showed as a result the nodal cilia are not positioned appropriately, which will disrupt the node's ability to initiate laterality formation. This figure illustrates in the top of the figure labeled A, staining of the cytoskeletal elements actin in green and tubulin in red. And so the two top pictures on the left show normal ZIG3 function, and the top two pictures on the right show when there is ZIG3 disruption. So those red dots represent the position of a basal body of an individual cilia in an individual nodal cell of the node. And then the lower images labeled B similarly have normal ZIG3 function on the left and abnormal ZIG3 function on the right, and show the position of the basal bodies of the cilia in red across the anterior-posterior axis. And so this effect on the anterior-posterior axis, in addition to the left-right axis, is why there can be a number of midline defects like neural tube defects in patients with ZIG3 anomalies. So in addition to interacting with nodal for the left-right axis that we saw back in that 2006 paper, and ZIG3 seems to have a ciliopathy of sorts due to abnormal positioning at the node for additional contribution to laterality anomalies. So overall, ZIG3 is a pretty interesting gene. And I mentioned I wasn't doing this story justice, and to really illustrate this, here's a non-comprehensive list of Dr. Ware's papers focused on just ZIG3 and heterotaxy, spanning from that 2002 up to just a few months ago. And this isn't addressing the work of many other researchers working on ZIG3, including one that we'll get to see a poster from this evening. And returning back to the big picture of laterality development, we know that things are set in motion for left-right axis formation long before we have any signs we can see. The first visible consequence of laterality is seen in the heart, which at least in part is because the heart is the first organ to form. This is one reason why cardiac involvement, often extremely complex congenital heart disease, is frequent in heterotaxy. However, we also know there are many patients with laterality disruption and no cardiac involvement. And these patients are really important to keep in mind because their anomalies might not be caught until later, and they may not be identified to have heterotaxy due to the misconception that heterotaxy patients must have cardiac involvement. For the moment, though, we're going to look at left-right axis as demonstrated by heart formation with another genetic example. These graphics come from a 2019 paper on DNAH5, a gene that results in primary ciliary dyskinesia in humans when you have biallelic disruption. And they studied the consequences of DNAH5 disruption on cardiac development in mice. So this is their graphic showing an overview of cardiac development. The top graphic labeled A shows some of the structures we've talked about in the embryo with the node in teal. And then the top right graphic labeled B is the node with the blue cells reflecting the monociliated cells that break left-right symmetry with their rotary function. And then these green monociliated cells are the sensory cells surrounding the node. And then this panel labeled C is a very brief overview of the nodal pathway and specifically calls out nodal lefty 1 and lefty 2 and PIDX2. And then across the bottom, labeled D through G, we see a timeline of cardiac formation, and you see where the heart develops progressive asymmetry. Now here we see the consequences of the biallelic DNAH5 disruption in the mouse embryos from that study. And they look specifically at nodal lefty 2 and PIDX2. And so since this figure is a little complicated, we'll orient ourselves. Down the left, we see A is focused on nodal, B is focused on lefty 2, and C is focused on PIDX2. Immediately to the right of that is a schematic of what view of the mouse embryo is being represented. And then immediately to the right of that are mice embryos that are representative of being heterozygous for DNAH5 loss of function, or what we would consider asymptomatic carriers. And then immediately to the right of that are 3 representative mouse embryos that have biallelic disruption, or what we would consider the primary ciliary dyskinesia. And you can see they all show different patterns of staining, and it's inconsistent across the different views. And then finally, all the way to the right are some graphs showing the patterns of expression for nodal, lefty 2, and PIDX2 in the embryos they assessed overall. And so for nodal, we have 2 sets of graphs representing the crown cells and then the lateral plate mesoderm. And then for lefty 2 and PIDX2, we just have the lateral plate mesoderm. So focusing just on the lateral plate mesoderm, these 3, you can see that it's color-coded to show that green is the proportion with no expression. Light blue is expression on the left. Gray is to show embryos with bilateral expression. And then the purple is those with expression on the right. And so this first row is embryos that have normal DNAH5. This second row is the heterozygous for DNAH5. And then this last one is the biallelic disruption, or the primary ciliary dyskinesia mice. And so for nodal, we can see that the DNAH5 biallelic disruption has increased incidence of no expression, some expression on the left, but then has bilateral and expression on the right. But the heterozygous, the asymptomatic carriers, also have a slightly different pattern of expression. They're more likely to have no expression. And for lefty 2, we similarly see that the primary ciliary dyskinesia mice are the only ones that have right or bilateral expression of lefty 2. And then the heterozygous ones maybe have a little bit more tendency to have left expression. And then for PIDX2, the DNAH5 disrupted mice are the only ones to have no expression, bilateral or right expression, and expression is on the left uniformly in the wild type and heterozygous mice. And so I think this is interesting to consider developmentally and clinically for two reasons. The first is that it reinforces that when signals are dependent on disrupted fluid flow in that node, that there will be an intrinsic randomness to how that abnormal flow will disrupt signaling, and that it can even be normal. The second is the embryos that we would consider asymptomatic carriers may show some predisposition to abnormal expression patterns or different expression patterns compared to those without, even though none have overtly abnormal expression. And I find that interesting because it supports something that we have evidence to suggest is true, which is burden of ciliary variants across a number of genes may predispose to laterality anomalies, even if they're just carriers, for multiple ciliopathies. And so while nothing is abnormal, it was different, and that makes it easier in my mind to imagine how a cumulative burden of variants may progressively predispose to laterality anomalies and ciliary dysfunction. And I think this is something that'll be important to patients with heterotaxy, as if we can identify ones with ciliopathies molecularly, we can potentially provide better supportive care. To do it, though, we have to be comfortable throwing out my 2002 Punnett squares out the window and putting our heads together in groups like this to come up with new, innovative solutions. But no pressure. And that brings us to our take-home messages. Laterality is established really early in development and relies heavily on modal and sensory ciliary function. There is intrinsic randomness and unpredictable elements to establishing laterality. And I think that this supports that in order to better understand and care for patients with heterotaxy, we need to come up with some new ways to analyze genomic information, particularly related to ciliary genes. Thank you for your attention.